Okay, so now, before we go into the more clinically oriented lectures, there's just one last thing I want to cover, and this is a bit of a catch-all lecture because it's viral genetics. But of course, since viruses are made of nucleic acids, and we're used to nucleic acids everywhere else, a lot of the genetics is going to be just the same as it is everywhere else. I mean, there's just the rules of molecular biology, and they apply to viruses as much as they apply to other things. But there are certain aspects of the genetics which are particularly significant in viruses. So I just want to go over these things to try and cover these things and put them in some kind of perspective. So understanding viral genetics has been important in understanding viral pathogenesis. It's been important in understanding the life cycles. A lot of what I've talked about has used mutants and variants and mutagenesis in order to follow them. It's been extremely useful for vaccine development, and a lot of our vaccines are based on changing these viruses in order to decrease their pathogenesis, slow down their life cycle, and enable us to develop vaccines. And it's also been important in understanding drug resistance and how you can screen for drug resistance and how you can develop drug resistance, etc. So the sort of usual things that you would think about for any pathogenic organism. But one of the things that I just want to point out with viruses is that there's a quote here from, I think John Holland, but it doesn't matter. But the DNA chromosomes, so our DNA chromosomes, require geologic time spans to change to the degree that RNA viruses can achieve in a single human generation. And that's because, in part, because our generation time is, 25 years or so, and a virus's generation time is 25 minutes or so. So a virus can achieve in a year what we can, in an hour, what we can achieve in 2 generations. So in terms of generation times, the viruses have got a huge number of generation times per year. So they just have a lot more offspring. And also, they have a lot more offspring per virus, as I pointed out. If you had 10 billion children, you'd be a bit upset about it, I think. So there's just huge numbers of viruses being replicated every few minutes in these the regular ones that we isolate. Many of them grow really well, and that's the kind of time that they would divide in. So it's just everything is speeded up in viruses. So that's one thing to remember. So they grow rapidly, that was the point. And as I say, a single particle produces a lot. But another problem, another reason that these RNA viruses seem to change so quickly, or viruses seem to change so quickly, is also to do with their nucleic acid. So the DNA viruses have access to proofreading because we proofread our DNA. So we have proofreading mechanisms that are particularly active at the time of DNA replication, but we also have regular patrolling DNA proofreading mechanisms that make sure that our DNA stays good from one week to the next and just keeps on patrolling the DNA and correcting any problems. And you've come across these various things, correcting for thymidine dimers and things like that, and proofreading. Now, proofreading takes a lot of coding potential. And the DNA viruses, by and large, have not taken the part of proofreading. They use ours. And so they're growing, if they're growing and replicating cells, our proofreading mechanisms are switched on. If they're growing in non-replicating cells, we have some proofreading switched on. But actually, it turns out, for example, herpes will actually switch on proofreading. So it actually uses our proofreading, but because it can grow in non-replicating cells, one of its proteins actually functions to trigger the mechanism. So, and again, I'm not going to quiz you on that, but my point is the DNA viruses have access to our proofreading and use it by and large. Pox viruses are probably a bit of an exception there, and they probably do their own. But so we've got, the DNA viruses have access to proofreading, so if errors are made, they can be repaired. So they tend to be reasonably constant. Now, they still have a high mutation rate because there's still a lot of errors and they're not being as efficiently controlled as we are. But they do reasonably well. But the RNA viruses don't have access to proofreading. We don't proofread our RNA, it's disposable. and they don't have access to proofreading, and they have never developed, apparently, the ability to proofread. And as I pointed out in the earlier lecture when I was talking about the properties of RNA viruses and why they have small genomes, part of it is because they can't proofread. So if you have and I think okay, I'll come back to that later. I think I got a slide on it. But anyway, RNA viruses, because they can't proofread, have a problem. And I already went over that at the beginning of the RNA strategy lecture. So the nature of the genome is really important, because if you're RNA, you're going to be much more error prone than DNA. And I've already pointed out that error proneness means that you're going to have small genomes, because if the inevitable rate due to the properties of the nucleotides are that you've got an error rate of 1 in 10,000, then if you have a genome that's much bigger than that, every genome is going to be mutated. Statistics say that if you've got an error rate of 1 in 10,000, on average, about 65% of your genomes will have a mutation in, and of the remainder, half will only have none and the other half will have 2. So at 1 in 10,000, if you have 10,000, you can probably get a reasonable number of OK genomes out, but if you're much larger, you have a problem. Also, whether it's segmented or non-segmented, and we'll come back to that in a minute. Okay, so I talked about genetic. So I want now to go into how you can change your genes. And there's 2 ways, really, how you can change your genome, rather. Mutation and recombination are 2 basic mechanisms. So I just want to go over mutation. It's basically the same as you've already heard before, but I just want to emphasize it from a virus's point of view. Okay, so I've already said there's an inevitable error rate, which I discussed a bit, but in that the bases that form the Watson-Crick base pairs, if you remember back to biochemistry, they probably maybe told you about tautomeric forms, in which there's a change in the electron distribution, just a temporary change, it just lasts for a very brief period of time and then it flips back again. But during that very brief period of time, where the change in the electron distribution, and don't learn this unless you feel you need to learn it for something else, but not for me. But the important thing is there's a change in electron distribution, and most of the time they spend it in the standard Watson-Crick form. But when they're in the other form, it happens that each of these behaves as if it was its opposite partner. So cytosine behaves as if it was thymine, adenine as if it was guanine. And therefore, if the base is in that form at the time the polymerase incorporates it, it will incorporate guanine when it meant to incorporate adenine. We immediately double check our DNA polymerase and bacterial DNA polymerase that copies the genome. They quickly double check to see whether the base that was just incorporated is a good base pair. And by the time they do the double check, if it wasn't good, it will have flipped back to its normal form and it will now be obvious it wasn't good. The RNA polymerases can't do that. So whatever the frequency of this is the basic frequency of mutation you can't get over. And as I say, it's about 1 in 10,000. So you always will get polymerase errors due to tautomers. And you also have the question of, can you ever make a perfect anything? And so polymerases probably also make errors just because polymerases make errors. But probably the major source of errors is actually this tautomerization. So, as I say, the mutation rates are usually much higher in RNA viruses because they lack proofreading. But that does raise one question, and which I need to discuss, because we have some viruses that they seem to change very little. After I've said all this, you'd think that viruses would just change in front of your eyes. But, the rabies virus vaccine, the vaccine that worked 10 years ago, the vaccine that worked 50 years ago, the vaccine that worked for Louis Pasteur would still work today, and it would work anywhere. Because the rabies hasn't changed sufficiently antigenically for it to be able to escape the neutralizing antibodies that vaccine raises. So rabies has remained constant enough that once you're vaccinated, that one vaccine will see everybody. So have mumps, measles, rubella. They're all RNA viruses. So vaccines for mumps, measles, and rubella, again, it's the same vaccine. It's been used for years. It's used everywhere, and the virus is not escaping that. So one of the questions is, why is that? And I tried to explain this with this figure, and I'm not sure it does it, but let's see if we can do it. The point is that with all these billions and billions of virus particles around, with different genomes in almost everyone because of this high mutation rate, the ones that infect best are the ones that will win this battle. They're the ones that will produce the most progeny. So the ones that infect best, are transmitted best, infect best, escape the immune system best, produce the highest number of progeny, are the ones that are going to take over the world. So they're going to climb to the top of the mountain. Okay. So you get to the top of the mountain and you're perfect, or as perfect as you can get, because any change that you make is going to make you less fit to compete with all these other viruses that are constantly trying to take you over. So as long as nothing in the big wide world changes, you're still going to remain perfect and you're going to remain at the top of the mountain. But as soon as you have a mutation in you, you're going to be right down here, and it's going to take a lot of struggle to get back up because you're always competing with this perfect virus. So the point is that for rabies, for mumps, measles and rubella, apparently everything that can change in here that would enable them to escape our neutralizing antibodies mean that they're no good in this fight. So you might be able to, if somebody has got mumps, measles, rubella, and you took all their respiratory secretions and you looked at every single virus particle in there, it's possible there might be one that could escape your neutralizing antibody. But it seems that if there is one that would escape it, it's not going to be very good at getting around, establishing an infection or anything else. And the only way it's going to get good is if it gets mutated back to this one, in which case our neutralizing antibody will neutralize it. So it just turns out that, that any change on that surface is apparently a negative change as far as this virus being able to win is concerned. So we do have constant RNA viruses, but in fact, if you were to look at each individual virus in anybody, you'd see that there are a lot of different viruses in there, but this overall antigenic serotype, it maintains those epitopes, and apparently any change in those epitopes means the virus doesn't compete very well. So it's a question of what the geneticists call fitness, and that's what I'm trying to show here. So we can get viruses that are constant, but that's because they're adapted. So one thing is, if something were to radically change, and I can't think of anything that would cause it for the ones that we're talking about, but if there was a radical change, you might be able to find viral variants. And we'll come to talk about rabies. Constant it is antigenically, actually in terms of nucleotide sequence, it varies quite a bit, and that's what we use for a lot of the epidemiology. So the origin of mutations, and you should know all this, there are spontaneous mutations, misincorporation of bases, etc. Physically induced mutations, UV light and X-rays. Now, one of the problems with UV light is it causes dimers. So if you remember back to biochemistry, you can get if there are adjacent thymidines in a DNA, the two thymidines can be linked together, and then they won't function properly in DNA replication. But we have a repair mechanism that will take those out and repair them and replace them with decent bases. There is no repair mechanism for uridine dimers. You get uridine dimers, but there's no repair mechanism. As I say, we don't repair RNA. So RNA viruses are very sensitive to UV light. So that's one of the things that they have relatively little in the way of protection on the outside because they've got relatively thin shells, and the RNA inside is very prone. So RNA viruses are very easily inactivated by UV light, and it's frequently used as part of maintaining a clean environment protocol. It doesn't remove all of them, but it at least reduces them. And then chemicals, as usual, will cause mutations, and it's no different for virus particles. And I'm not going to go into any of those. You get the standard types of mutation. You can get point mutations where one base pairs for another. You can get insertions and deletions, just as you get with regular genetics. I mean, it's no different. So you can get all these kind of mutations, but they just occur very rapidly and very frequently, and you've got a huge number of virus particles, and if one of them has a mutation that's going to make it really fit and able to compete with all the others, it can take over very quickly. So what do we look for when we're looking for the genetics of the virus? You can't poke it, you can't run an ECG on it. You can't do anything that asks it anything much. So what do we look for? So what are the phenotypic changes that we will look for in a virus and that's the observed properties of an organism. So the genotypic changes are changes in the genome, and then the question is, what do we see as a result that we can actually measure to show there's been a change? Well, one thing that is commonly used in virology is the use of conditional lethal mutants. And that means that these mutants will grow under some conditions, but other conditions are lethal to them and they won't grow. So that means you can grow the virus under certain conditions, and then you can put it in other conditions and say, will it grow here or won't it? So, for example, if you take a wild-type virus and put it into tissue culture, it will probably grow at 31 degrees, and it will probably grow at 37 degrees. It will probably grow better at 37 degrees. After all, these are human viruses that you're interested in. They probably will grow better. But you can get some temperature-sensitive mutants that will grow at 31 degrees, but they won't grow at 37 degrees. So that's a condition which is lethal to them, is 37. So temperature-sensitive mutants are a type of conditional lethal mutant. And that's frequently because they've got a mutation that leads to an altered protein, and that altered protein will no longer function at 37 degrees. These are very useful because, for one thing, it's useful in vaccines. So, for example, the live flu vaccine has got conditional lethal temperature-sensitive mutations in it so that it will grow in the very upper respiratory tract, but it doesn't like getting down lower because the temperatures are getting higher and it doesn't grow at all well. So if you make a virus that will only grow close to the outside of the upper respiratory tract, then it remains in your nose and it's not much of a problem. And what you're worried about with these viruses is if they get down into the lower respiratory tract. So temperature-sensitive mutants are very useful. You can also get host-range mutants that will grow in some cells and not others. And when we used the live polio vaccine, which was used for many, many years, the live polio vaccine had got a mutant which meant that the vaccine virus would not grow in nervous system cells, in nerve cells. So the regular virus will grow in both types of cells. The mutant wouldn't. So that was a safety mechanism. So you make these viruses so that they don't grow very well and they don't go for the target cells that cause the real problem. Polio growing in the gut isn't a problem to us at all. We don't even know we've got it going in the gut. But once it gets into the nervous system, we do have a problem. So we are constantly searching for these kinds of phenotypes. They're very useful. I talked to you about, in one of the early lectures, about how we measure viruses, the number, by looking at how many plaques we can form, because one infectious virus, if you do the experiment properly, will give rise to one plaque. Sometimes you see alterations in plaque size, and the alteration itself doesn't tell you anything. They can be small or large. All it tells you is that the virus that started that plaque, and or its progeny that formed that plaque, have got something different about their genome because they're causing a different size plaque. But then if you take these variants and examine them, you often find they've got altered pathogenicity. So again, they're useful for telling you what's pathogenic and what's not, and for trying to develop live vaccines, because in most cases, live vaccines have got big advantages. They have some disadvantages, but they've got some big advantages. One is that you get cell-mediated immunity. Another is that you get IgA. So drug resistance is another important phenotypic change, and unfortunately with influenza we're starting to see more and more of that. For years it looked like we were pretty safe, and now we're seeing problems. And you can get enzyme deficient mutants, and I've already pointed those out with herpes. It has a thymidine kinase that is used to make the precursors for DNA synthesis. That is not necessary for herpes virus in rapidly growing cells which are making plenty of their own thymidine kinase and plenty of their own deoxynucleotides. But if you're in a non-dividing cell, then you need it. So it needs it under certain circumstances. So those are enzyme deficient mutants, and they're useful, and we'll come up to one use of them in a minute. You can get hot mutants which grow better at a high temperature than does the wild-type virus. Those can often be more pathogenic because part of our antiviral response is fever. And a lot of these viruses don't grow so well under high temperature conditions. So fever actually does help deal with these viruses. But if you've got a mutant that likes fever, then you've got more of a problem. And we also get attenuated mutants that have very mild symptoms. And we've already talked about mechanisms of attenuating: temperature sensitivity, host range response, etc. And as I've said, they're useful for vaccine development. And also, if we know why they're attenuated, it tells us which proteins are important in pathogenesis. And those are ones that we can maybe target better than others. So that's just a brief let's just put the common sense together as far as it goes for for the mutations. What about recombination? Now, recombination in viruses is a bit weird for many of them, and we'll go into it. So my definition of recombination is exchange of information between two genomes. So you have two genomes at the beginning, and at the end, you have a genome which is not like either of them, and has got properties of both. So they've exchanged information. And so the question is, how do you do it? There's the classic recombination, which very superficially, I'm just showing it as a thing whereby you've got your two genomes, the pink and the purple, pink and the blue, and then there's a crossover event, and you exchange genetic material with them. And so here you've now got, as I say, two new types of genomes, which have got something from one of them and something from the other of them. And so that's classical recombination, and the DNA viruses can do that. They've got recombination mechanisms, and the recombination mechanisms are basically heavily based on ours. They snaffle our stuff again. So you get a lot of recombination in the DNA viruses. The mechanisms are available for pirating, and they pirate them. So it's common in the DNA viruses. And again, pox virus is cytoplasmic, so it doesn't have access to that, but it does have its own mechanisms for recombination. So it's enormous, and it has to be enormous considering what it's doing for itself. So, whoops, let me go back. Okay, so that's common in DNA viruses. But what about the RNA viruses? They don't have access to recombination. So what are they going to do? Not to DNA recombination. And so the simplest one that quite a lot of these viruses use is something called copy choice, or a variation of it. But I'm just going to discuss what copy choice means. Basically, here you've got two positive sense RNA viruses. This one has got a good sequence here and a good sequence here, but this one has got a mutant which they will kill it here, and a mutation that will kill it here. So neither of these is going to be able to survive in non-permissive conditions. But if you put both of them in the same cell, then the RNA polymerase that starts off by copying this one will make a negative strand, which will have the good sequence here. If this polymerase then takes the RNA that it's growing with it and aligns on another strand at the exact same place, because of course there's a sequence here that will be hybridizable to strand 2, if this falls off and hybridizes to strand 2, which is called template switching, this will all basically match strand 2, and then it will carry on here and copy strand 2. And so now what it's got is it's got the good mutation from the right-hand side of strand 1 and the good sequence from the left-hand side of strand 2. And what you've now got is a recombinant strand. So it's just by copy choice. This is negative strand, but of course it's copied into positive strand. And now you get a recombinate positive strand, and that can go on and make good proteins, and you can get it packaged into virus particles, and you've got recombination. So this happens in poliovirus, it happens in coronaviruses, it happens in quite a lot of viruses. That or a variant on it, but this is the simple one. So it occurs in the positive strand viruses. As I say, there are variants, some of which are very efficient at causing copy choice type of recombination. And I'm not going to go into those. The reason that you can get variants that are more efficient is because some of the other positive sense RNA viruses have got slight differences in the mechanism of how they make their RNA. And if you take advantage of that, you can actually get huge amounts of recombination. So what about, that was the positive sense. What about the negative sense RNA viruses? Now, we have a problem with the negative sense RNA viruses because I've said the negative sense RNA viruses, as they copy their genome, they coat it with nuclear capsid. And it seems that this nuclear capsid stops copy choice going on. It's very difficult for this polymerase to fall off and find the right place to start on the next strand. So it doesn't seem to ever do it. We have not been successful in isolating recombinants of that kind from these viruses. So what can it do? It just has a big problem because it can't do copy choice and it can't do the DNA type. So you don't see a lot of recombination in the non-segmented negative strand viruses because their genome is always coated with protein and it's not open to DNA type recombination, it's not open to copy choice type recombination. So they remain much more stable in terms of recombination than do the positive sense RNA viruses by and large. So as I say, it just can't find the right place and nothing, you don't get it. And I've already mentioned this. So let me just check something. Okay. So while we're talking about recombination, what are some of the uses of this sort of recombination? And one of the things is you can map where genes are by recombination frequency. This was important a long time ago because the idea is the closer two genes are together, the less likely you are to get a recombination event between them. If you've got huge long stretches, then the likelihood of recombination event can occur over a huge stretch is much higher than if it's just a very short stretch. These days, though, sequencing is so good that really we don't need to map genes by recombination frequency anymore because we can sequence all of these viruses in an afternoon if you've got the right equipment and enough money. So mapping by recombination frequency largely is historic. You can also map by marker rescue, and that's historic, but it brings up a mechanism that I want to discuss. So let me just go through what marker rescue was, or is. Okay, here we've got a mutant herpes virus. We can grow it in tissue culture cells because the mutation is in thymidine kinase, and so it can manage to grow with that, but it doesn't have thymidine kinase activity. So the question is, where in the genome is thymidine kinase mapped? Now, I've given the secret away here. I'm not sure that's actually where it is. This is just where I parked it. But what you can do is you can chop herpes virus DNA up into bits with restriction enzymes, purify the individual bits on their size, extract them, and add them back. And then recombination can allow the right piece will line up by homologous base pairing, and you'll get homologous recombination, and you'll get this inserted into the mutant HSV, and it will become a wild-type herpes virus with a fully functional thymidine kinase. So that's called marker rescue because you rescue this phenotype by adding the right piece of DNA, so it's a marker for which is the right piece of DNA. So it was heavily used, but as I say, these days with sequencing, most of this is going by the board, although you'll see the terms marker rescue appear in the books periodically. Well, they're still in the books. But it has got, this same technique has been used for developing vaccines and also for developing gene transfer, viruses for gene transfer. So here's one that's actually used. So here we've got vaccinia virus, which also codes for a thymidine kinase in order to get lots of DNA for itself, lots of DNA nucleotides for itself. And what you can do is you can isolate the fragment of vaccinia virus that contains the thymidine kinase. And actually, because it's so huge, it took a very long time for that to be sequenced. So marker rescue was useful for isolating it. And then inside that, you take and insert a rabies virus glycoprotein, okay, with an appropriate promoter and everything else. So you insert the gene for rabies virus glycoprotein in here, and that's the surface glycoprotein, remember. The G protein is the one that, if you've got neutralizing antibodies to it, will protect you. The homologous recombination, since it's got thymidine kinase sequences on either side, you still get thymidine kinase, so it will insert this into the gene. The vaccinia virus won't now have thymidine kinase because this gene is ruined by having rabies virus inserted in the middle of it. But the rabies virus gene can now be expressed, and you'll make rabies virus glycoproteins. And they'll be inserted in your host cell membrane, and your immune system will see them, and you'll develop antibodies and cell-mediated immune to this. So it's... Now, we don't have this virus. This is not the basis of the rabies vaccine that we use, but it is the basis of the rabies vaccine that raccoons use. Now, if you didn't know that raccoons went to a rabies vaccination clinic, you're learning. We have a lot of rabies in raccoons on the East Coast and in foxes. And it's getting to be a major problem in some areas. So what they're trying to do is reduce the load of rabid raccoons and rabid foxes as well. So what they do, and this is going on in West Virginia, Tennessee, various other states, what they do is they drop these baits, which apparently smell really yummy to a raccoon, but smell pretty disgusting to humans. But raccoons will eat anything, apparently, particularly if it's nice and tasty, which apparently it is. So the raccoons pick up the bait and start eating it, and inside it is a little packet, and when they puncture the packet, they get this virus. And it's a vaccination, and they're protected against rabies. So this is currently in use. Okay? Any questions on that? So, as I say, nothing much is here new. It's just maybe a slightly different way of emphasizing it. Okay, now there's another important thing we said the negative sense, the positive sense RNA viruses have copy choice by and large, or some kind of variant on it. The negative one, the non-segmented negative strands can't do anything. And the segmented negative strands can't use copy choice, but the segmented negative strand viruses have an option, and the segmented double-strand viruses have an option, and that is something called reassortment, which in a way is another kind of recombination, but it's rather different, so it has its own name. So here we're taking two viruses which have a segmented genome. In this case, they have two segments. So the two viruses get into the cell, and they make lots of copies of their small segment, both pink and blue, and lots of copies of their large segment, both pink and blue. When they come to assemble new virus particles, it's just random as to whether you get two pink, two blue, or one pink and one blue. And so it's extremely efficient. It's just basically, there's all these small segments out in the cytoplasm, and as far as packaging goes, there's no clue as to which virus they came from. They just all mix in the cytoplasm. So it's highly efficient. So these segmented RNA viruses with reassortments can exchange segments very easily. And so one of the things is that, as we'll come to see when we talk about flu, that's one of the bases for flu variation. And that's what we're having this year. We've got a reassortant virus that's causing the pandemic, because it can suddenly pick up a segment from somewhere that it shouldn't have done so. And also it's used for making vaccines. So, as I say, it's a form of recombination. It's very efficient, only in segmented viruses, and it's in the newer vaccines, such as the FluMist vaccine and the rotavirus, one of the newer rotavirus vaccines. So if you look at this is the nasal live flu vaccine. This is a slide of showing how it was originally done. So it was a cold-adapted virus, so in other words, it grew better in your upper respiratory tract than down below. It was because it prefers the cold. It's temperature sensitive, so it hates the heat. So it really doesn't want to go any further down anyway. It's attenuated, so that even if it got further down, it wouldn't cause much in the way of disease. It's live, so you get a mucosal response, mucosal immunity, and a cell-mediated immunity, as well as IgG. It's delivered intranasally, which, from the point of view of little kids, has sometimes got its advantage. And it was finally approved after many, many years of trying in 2003. So this is a reassortant vaccine. So what they do is they're their quotes donor virus has got all these segments in it, 2 of which code for the surface proteins. And then you get the nasty virus, which has got all these segments in it, 2 of which code for the surface proteins. And then what you do is you put them together and let them get on with it. And when you look at the outcome, what you isolate very carefully are those viruses which have got all the grey genomes, with the exception that the 2 surface protein, the grey segments, with the exception that the 2 surface protein segments are coming from the new virus. So you can very quickly make a new virus which has already basically been tested, because all of these have been tested for making sure that they're giving you a virus which is attenuated, not going to cause problems, etc. But now you can slot in these 2 new segments just by reassortance. So that's how that virus is basically put together. So we have a reassortant vaccine for rotaviruses as well, because, of course, they're double-stranded RNA, but they do the same thing, where there's a mixture of bovine and human things in order to make it safe. The other rotavirus vaccine that's currently available is just highly attenuated. But again, this is the whole principle of understanding the genetics and trying to assay for all these things. And then the poor old non-segmented negative strand viruses have got no classical recombination, they've got no copy choice, they've got no reassortance, and they've got the least ability to exchange genetic material. But they can still change pretty rapidly because they've got this business about pretty sloppy RNA synthesis because of all the proofreading problems.